Good morning, everyone. Thank you for attending to my presentation. Thank you also, Jaime, for the introduction. So basically today I'm going to be talking about the model application, but also improvement of agroecosystem models that can serve as a tool for the design of crop diversification in different spatial arrangements at the landscape scale. So first, I'll just give you a very brief introduction about agriculture in Germany, which I'm sure you are mostly aware of. But agriculture in Germany, it's a quite strong sector. It's also one of the largest in Europe, and it's characterized by large farms, which have been increasing even more over the last years. And they are also characterized by highly mechanized, with high use of machinery and also high use of inputs such as fertilizer and pesticide applications. Also, over the last decades, it's been identified that the crop rotations also have become more simplified over time. And despite the high productivity of these agricultural systems, also it has come with different consequences to the environment. For example, the fertilizer leaching to the water bodies, which is a major concern, but also the increased risk of greenhouse gas release to the atmosphere, particularly for nitrous oxide, which typically increases with the application of fertilizer, of nitrogen fertilizer, but also a high concern on the decline of biodiversity as we have increased the intensification in agriculture and also the larger field sizes. And also an increased yield risk due to these simplified crop rotations, but also due to climate change, which is also expected to continue increasing in the future. So as an option to all of these problems, to cope with all of these problems, we have identified that crop rotation and smaller field sizes, they can be an option to cope with these problems. There have been a series of reviews that have been, and research that has been published over the last years, decades on this. I mean, it's not a completely new topic. But these recent reviews, they have identified that crop diversification resulted in increased productivity typically, and increased biodiversity, and also an improvement in ecosystem services such as the improvement on water quality, also pest and disease control, and soil quality. Also here to the right, you can see another paper review by Tamburini in 2020. Which he basically looked for the responses in the system to any diversification practice, and what was the effect of those in different factors of delivery of ecosystem services, but also in terms of biodiversity. And he found that it was mostly a positive response in most of the studied factors. So a positive response in soil fertility, crop yield, but also in nutrient cycling, also improved pest control and increase on biodiversity and pollination. Also in terms of smaller field sizes, so there was also another review that basically you can expect that because with the increasing field size, there is a loss of biodiversity in the fields. So it's expected that with in other past research has shown that when you have smaller fields, typically there's a higher biodiversity within the fields. And as the field size increases, then the biodiversity tends to decrease. Also, another important thing that was studied for this paper, it was the gross margin. So the gross margin that may decrease when you reduce the field sizes. But they also mentioned that with alleviative measures, this crop margin can be, the risk of loss of gross margin, it can be improved with these alleviative measures. So in addition, we have also agroecosystem models, which can serve as a tool for the optimization of these systems. Basically, they are mathematical equations that represent, they are a set of mathematical equations that represent important plant processes and their interaction with the environment. So here on the right, you can see a very rough diagram of them. So basically, we have the the weather variables, which are typically solar radiation, temperature, daily rainfall. And then we have a set of crop parameters that define the crop type and also the different crop cultivars. Then we have the field operations. So for example, fertilizer application, when it occurs, the amount that it's applied, and also the type of fertilizer that is used. Also, other field operations can be also if irrigation is applied, so how much and what is the frequency of them. And then also the soil properties by layer, which is also a very important input data for the models. And basically, we need the soil properties by layer in terms of soil texture, pH, soil carbon, and water properties of these soils. So these models typically they run in a daily scale, and the minimum simulation unit is 1 square meter. Typically, it's able to, these agroecosystem models are able to simulate also the crop development, growth, yield, and resource uptake by the plant, and we are able to quantify certain ecosystem services with them, which I'm going to mention later. Also, they can be very useful for model of scaling, so going from local to the regional scale to try to study these impacts, and they can also, for this, serve as an exploration tool also to help to provide information on the design of field experimentation tools. So it's two tools that can go on hand. So the agroecosystem model that we use is within the Simplex modeling framework. So the Simplex modeling framework basically is comprised of different sync components. And one sync component basically it's a small simulation unit that represents an important process in terms of crop growth and development. And then an agroecosystem model, basically it's a combination of these sync components. And the combination of the sync components, they are gonna depend on the complexity of the simulation, but also they are gonna depend on the availability of observations and the quality of the observed data. Here to the left, you can see just a diagram of the three main components. So we basically use the weather as an input to the model. Then we have the different crop representation and crop processes, and then we have also the soil processes occurring. And basically, each of these boxes, it's a sync component that it's used. So, for example, we have in terms of phenology, it's simulated with lintel phenology, which is mostly affected by the accumulation of degree days. Then we have the growth, which in this case, there's other sync components. For example, we are using actually lintel 5, but these components that are mentioned in this diagram are also available. So basically, we have that we simulate the lintel biomass. Lintel biomass basically simulates the total daily biomass accumulated based on the radiation use efficiency and water and nutrient factors. And then there is also another sync component that simulates the partitioning of this total biomass into the different crop organs. And similarly, in the soils, there's also a sync component to simulate the dynamics of the root growth over the soil profiles, and also the nutrient and the water dynamics that affect ultimately the crop growth. So some of the research questions that we are trying to answer, and I'm going to try to answer during this presentation, it's mostly 3 of them. So we would like to know first to what extent the agroecosystem models have been used for the crop diversification design at field and landscape scale by considering different soil heterogeneities. Also, we would like to know what are the model requirements for this purpose. And as a third question, and in this third part, I'm going to show you some of the data that we have collected so far. And the research question is on whether the model is capable of simulating crop responses to within field heterogeneities. So for the first two questions, we conducted a paper review that we submitted early this year, and I'm going to show you some of the results now. So we define crop diversification first at the local scale. Basically, there's you have a spatial crop diversification that it can be as simple as genetic diversity, which is same crops but different cultivars in the same field, and you can have more complex ones, such as, for example, agroforestry, which is the combination of trees with crops growing in the same field. And depending on the mixture, there can be, minimum 2 crops or crop species that are mixed up to a undefined number. And then for temporal crop rotations, typically we have basically crop rotations, so sequential growing of one crop after the other in the same field. So for models that have been used for crop diversification at the field scale, these models, they have been mostly applied for evaluation rather than design of the systems. The minimum field unit, as mentioned before, it was also 1 square meter in the case of these agroecosystem models. And in this table below, you can see some of the examples. So for the spatial scale, there is about 7 agroecosystem models that are able to simulate spatial crop diversification. But typically, the number of crop combinations that are possible is just 2 at once. Some of the advantages of these models is that they have simplified approaches where they express the crop interactions mostly in terms of light, nutrients, and water competition. But then some limitations are that they have, for example, these restricted crop combinations that is just 2 at once. And typically they comprise cereal, legume combinations for specific crops, and also cereal and cereal combinations. And typically these combinations are for the more common crop combinations that you can find within intercropping systems. As for temporal, there's many more crops available, so typically between 13 to 70 crops. Sorry. And there are different models, more than 8 models that are able to simulate crop rotations. Because, of course, at the beginning, all of these agroecosystem crops models, were created to simulate sole crops and not intercropping, for example. Some of the advantages is that they already are well advanced for the simulation of major cereals, legumes, and also root and tuber crops. But some of the limitations that they have is that the crop parameterization, for example, for marginal crops or for niche crops, it's quite limited due to the availability of data to set up the models for these new crops. But also soil and soil carbon and nitrogen dynamics, multi-year dynamics, sometimes in the long term. For some models, they are not as stable. So there's also an improvement on this part. As for the modeling of crop diversification at the landscape scale, there are basically in this part we try to we define the landscape as approaches that attempt to design diversified cropping system in an area encompassing at least several cropping fields or farms. And basically the spatial diversification at the landscape scale it's given by two factors, so landscape configuration and landscape composition. And you can see this diagram here on the right. So basically, the landscape the landscape composition, basically you can have in this example in the in number A, well in letter A, we have, for example, in this square we have two crops. So if you want to increase the compositional heterogeneity in this field, then there is an increase on the number of crops within the same field, and in this way the compositional heterogeneity increases. In terms of configurational heterogeneity, it increases when the field sizes within the area are reduced. So basically, from having four, let's say, fields, in this case, to going indeed to have more than 15, let's say, field size, different fields. So basically, crop diversification at the landscape scale is increased by these two measures. And typically at the landscape scale, in agroecosystem models are not typically used. What it's used is decision-making frameworks with optimizations on agronomic, economic, and ecological terms, depending on the type of model and the research question. The minimum scale that it's typically optimized is the field plot, and crop rotations are optimized within the boundaries of individual farms. And then the crop configuration and composition basically is gonna depend on the arrangement of the crop rotations and their spatial placement. Some examples for these frameworks is crop rotate, rotor, and life cycle assessment. And fruit fungi, I think it also falls within this category. They are typically not dynamic, and they have simplified crop assumptions. As for the provision and regulation of ecosystem services, there's many of these that they can be already simulated, but they depend on the model. So for example, all the models are able to simulate, for example, food and health, because they simulate the biomass and also the grain yields, in terms also of vegetable fiber and fuel. They can be also derived from the biomass that is simulated. Then typically the models also account for nitrogen dynamics, so nitrogen related ecosystem services, they can also be quantified using the model. So for example, rotation and of course nitrogen leaching. Also for pesticide fate, not all the models are able to simulate pesticide fate, but for example, the STIX model is able to simulate this. As for soil erosion and soil runoff, also soil erosion is considered in some of the models. Right now, there's a model improvement going on in our group for a student that is working in this part of including the soil erosion by water into the SIMPLACE modeling framework. And also we are able to simulate already air runoff, and many models are able to simulate air runoff dynamics. Also in terms of greenhouse gases regulation, so the many models are also able to simulate the dynamics of nitrous oxide emissions, and also soil carbon sequestration in the long term. Some of the required modeling capabilities for newly diversified arrangements in heterogeneous fields and landscapes that we identified, where the inclusion of, that we need the inclusion of crop species and cultivars, for example, to improve the part of the local diversification. Also, crop interactions are important. So have a good representation of the crop interaction in terms when you have crop mixtures. Also, the crop responses to within field heterogeneities. If it's whether the models are able to simulate these crop responses, and also the it's important. It will be important in the future also to at the impact of also diversified field arrangements in biotic stressors, because we know that with crop diversification, there's a possibility to improve pest management, for example, in the fields. So to be able to quantify this, and also in terms of quantification of the delivery of ecosystem services, and also biodiversity, which are main also topics for us in the part of the model improvement. So one framework that we proposed in this paper review that we submitted, it was to basically a framework for the design of diversified cropping fields. So in the first part, we have a static part, basically that we do the field classification depending on the physical properties, chemical properties, historical yield maps, and also the crop selection range based on the patchCROP, new crops, and different restrictions. Then we have a dynamic part, where we use the agroecosystem models. So basically, we try to run the different set of crops, field sizes, and field geometries, and then we try to optimize these three factors based on the evaluation of trade-offs in terms of the delivery of ecosystem services, in terms of provision, regulation of ecosystem services, and also in terms of biodiversity. So as a last part of the research questions that I have for you today, one other that is very important for us is the model capable of simulating crop responses within field heterogeneities. And this is quite important for us because we have seen that there's very large heterogeneities in the patchCROP field experiment, which is our main source of input data. So I'm going to talk to you a little bit about the experiment and all of the soil information that we have been collecting, and also a model test that we did with 3 crops to see the different crop responses to this within field heterogeneities and different weather conditions. So, well, the patchCROP, I'm sure you all have heard of it till today, but just to give you a bit refreshment on it, well, this experiment, it's a non-farm field experiment that it's located in Eastern Brandenburg in Tempelberg, and basically we have an arrangement of 30 patches, so you can see here the full field, and then we have an arrangement of these 30 patches, and each of these patches measures 72 by 72 meters, which is roughly 0.5 hectares, which is the minimum field unit that it can be managed with the current machinery. And then these patches are divided in field quadrants. So there's a quadrant where the crop growth measurements are taken, another one where the soil measurements are taken, and the third one where the biodiversity measurements are taken. And then there's a fourth one that is available for new research questions. Then in addition to this, also we have the different reference patches around this field. So last year, for example, we had the maize soil crop field in this area here. So it was a large soil cropping system that we try to use also as comparison to see how this patchCROP arrangement dynamics compare also to the soil cropping. The field is divided also in 2 yield potential zones. So basically the high and low yield potential zone, this division was based on a cluster analysis made by Marco Donat in another project. And basically here to the top, we have the high yield potential patches. So it will be this one here on the first one in the left, then the first 5 set of crops in this cluster here, and then these 5 set of crops in this part. They belong to the high yield crop rotation, and then the rest they belong to the low yield crop rotation. And then there's different management windows. So the management window 1, which is basically standard management, so business as usual. Then there's the second window of management, which is reduced management, so trying to reduce the application of pesticides, particularly for weed management. And then we have the third window in pink here, which basically it's the same reduced management with the addition of the flower strips. There's a site-specific crop rotation, so we assign it. So there's one for the high yield crop rotation, and then there's one for the low yield crop rotation. And typically, we expect that the high yield crop rotation, it has a higher nutrient demand, and therefore it was placed on this high yield potential zone. Some of the observations that we use for the initial model calibration are the ones from window 1 so far, because those are the ones that we are able to simulate with the model. Because in the model, so far we don't have any inclusion of for example, incidence of weed or pest, so it will not be possible at the moment to simulate the dynamics of the other two windows. But in the future, we hope to be able to include also this part of the management within the model. So some of the data that we use for the model calibration is the daily weather data, the crop phenology, so the important stages of crop phenology, also the intermediate biomass that it's collected over the season about 4 times during the season, the leaf area index, which is collected weekly up to during the vegetative stage, and then during the reproductive stage around 2 times a month. Then we also use the crop yields, the crop final yield, and the initial soil nitrogen and water condition, and the soil properties, which, as I mentioned before, it's well as all of the previously mentioned data, it's also very important in our model simulations. And for this, for this part of the soil properties, we have been conducting an extensive soil sampling campaign at the patchCROP experiment. So far, we have been able to collect soil data from up to 1 meter depth from all of these patches that are here in red. So we have collected already information on 11 patches, and we have a quite good distribution now with around 5 patches in the high yield crop rotation and 6 patches on the low yield crop rotation. Also, some of the characteristics that we have been able to collect during this campaign, it's so for the auger points, so we do some analysis in the field and others that we do at the lab here at the University of Bonn and also in Jülich. So some of the information we have, it's the color fresh, the gravel content, the textural manual reading, according to the German classification, also the calcium carbonate content and models. And then from these soil augers that we collect from the field, we then take as a selected amount that we bring to the university to conduct further analysis. So to try to identify the soil particle analysis for each texture class, also the total carbon and nitrogen content, the pH, and the extractable P&K. And all of this information, it can serve us as to have a very detailed input on the soil properties, and to be able to try to simulate the crop responses to all of these heterogeneities that we see in the field. So some of the early findings we had, it was that, for example, for soil texture and for total carbon, if you can see these 2 graphs here on the right, basically and this is just for the first 3 patches that we sampled. This was in the first campaign. So we did 3 patches, in total 48 auger points, and then these results are from some of these selected auger points we took to the University of Bonn. So for the topsoil, you can see that typically the textural classes, they are mostly within the sandy textural classes. And they are quite, let's say, it's more or less homogeneous in the topsoil in regards to this. So for example, for the patch 12, there's 2 soil textural classes. And then for the patch 102, which is a low yield potential patch, and 114 that are low yield potential patches. Also, there's also 1 textural class for each of them. Then, and we also see that, for example, the SU2 and the SS, they have a higher textural, higher sand content, sorry. Also for the total carbon, typically we saw that for the high yield potential patches, the total carbon, it was slightly higher than for the low yield potential patches. Also, another thing that we observed, it was that the soil textural classes in the subsoil, they were more heterogeneous. And also the total carbon content, it decreased drastically from the top layer to the subsoil layer. So these ones, we treat them as the same when we use this data as an input to the crop models. Also, another interesting characteristic that we found, it was the distribution of the sandy layer. So in the top soil, typically you have dominantly soil textural classes, but then as you go deeper, then there's the appearance of a loamy layer. And this loamy layer, it's quite important for us to identify because it affects the soil water retention characteristics. And you can see from these graphs that it was quite variable. So if we look here at this top right graph, this is the average depth of the appearance of the sandy layer, meaning that you have this sandy layer, and then after this, you're gonna have a loamy layer. And this is presented for the high and low yield potential patches. So P12, P19 it's each of the patches, and then for the low, we have the same order. So patch 76, 89, all of these that belong to the low yield potential patches. So you see that there's a quite high variability first on this appearance of the loamy layer. Also, it varies even within the patch. So we collected these augers in the soil and the yield quadrants, so we can see that even within the patch, there's a difference in the appearance of the loamy layer, and also there's a large variability. The error bars represent the standard deviation of all of the augering measurements within the quadrants. Also, if you look at the full distribution of the depth of the sandy layer, you can see that it tends to be quite variable, regardless of being high or being a low yield potential patch. But then in average, when you look at these values in average, typically you see that in the high yield potential patches, the appearance of this loamy layer, it tends to occur at a shallower depth than in the low yield potential. But you see also that, I mean, the variability, there's still variability within these measurements. Also within this campaign, we also identified that some of these soil augers that we have sampled, some of them, they have a full sandy profile. So for example, and it mostly happens in the low yield potential patches that you can see here. So for example, in patch 76, 89, and 95, 15 out of the 16 soil augers that we collected, they had these fully sandy soil profiles. Then patch 89, it had 10 out of 16. And then in patch 95, there were all of them basically. But then we have also exceptions, because even in the high yield potential patches, we saw also one of the patches that had very frequently fully sandy soil profiles. One particularity of this high yield potential patch is that it's within a small depression in the field. So it's possible that it's because this one, it has a higher accumulation of colluvial material. So once we had all of this information that we have collected for the soils, we wanted to conduct just a modeling test to try to see how some of the crops that we have in these crop rotations are affected by these soil characteristics, and also if the magnitude of these responses, it varied between among the crops that we use. For this, we selected two contrasting yield potential soils, soil augurs, that you can see the description here. So the 12, it belongs to a high yield potential patch, and the 114, it belongs to the low yield potential patch. And you see that the sand content in the high yield potential patch, well, it's still high, but it's lower than the one that you see in the 114. So in the 114, you see that they have more than 85% sand content, while in the patch 12, the sand content in the bilayer, it varies from around 60 to 68.7%. And also in terms of total carbon, well, for this particular case, actually the low yield potential patch had a higher total carbon, although generally we saw that the low yield potential patches, generally we saw that the high yield potential patches actually had a slightly higher carbon. But in general, you can see that the total carbon in the field, it's quite low, it's less than 1%. And then it's even lower in the subsoil. So here just some summarized results from them. So you can see here well, and I forgot to mention that this simulation was conducted for a 10-year period. So here you can see the summary of these 10 years of simulations for grain maize, soybean, and winter wheat. And this is the top biomass and grain yield responses depending on the soil. So you have the 12 here, high yield potential, and the 114 here, low yield potential. So you can see that typically the high yield potential tends to accumulate more biomass and also more grain yield by the end of the season for the three crops. And then if you see, as for the percent differences, we can see that also the lower differences were observed in the grain maize, but then also the soybean and the winter wheat, they also tend to show even larger differences. But you can see that the differences between one soil and the other, they tend to range between 20 to 40%. So it's it's for sure that these soil characteristics, they affect differently the crops. Also, in terms of maximum leaf area index, simulated maximum leaf area index, also we can see a similar pattern. I mean, we have a reduced growth in the low yield potential patches, in the low yield potential patch for grain maize, soybean, and for winter wheat. It's a similar pattern within all of the crops. And if we look at the 10-year differences for maize, for soybean, and for wheat, we see that the asterisk represents when the rainfall, when we had high seasonal rainfall, and the red asterisk represents when we had a lower seasonal rainfall. And basically, you can see that for maize, there's differences between one. So the dark line is the high yield potential patch, and the green line is the low yield potential patch. So you can see that, over the time series, you can see that there's some differences. Most of the time, you have a low lower yield with the low yield potential patches. But then if you look at the soybean, for example, and the winter wheat, these differences between the soil, they are even more drastic. And then in some cases, so for example, in the maize, when there was an event of low seasonal rainfall, then the yields were similar between these two soils, but then when there's high rainfall, it seems to be more, the crop seems to be more responsive because there's more accumulation of the water within the soil profile. But then soybean, the differences in soybean grain yield, it seems to be, when you have less rainfall during the season, then the reduction on either soybean or in winter wheat, there tend to be more marked differences in the low yield potential soils. So for some of the findings on this last question, so the patches show within and among field heterogeneities, mostly regarding to the appearance of the loamy layer in the subsoil. So the top layers tend to be quite heterogeneous, quite homogeneous, on the top, but then in the lower, we see all of these heterogeneities in terms of the soil texture, mostly that affect the soil water retention. Also, the soil carbon, it tends to be higher in the top layer than in the subsoils, and crops respond in different magnitude to the soil type and the weather conditions, which is an important thing that we have to take into consideration when optimizing the spatial crop diversification. And then, because also we have seen that this collection of this soil information so far, it's been done manually, which is quite intensive. So we hope to that in the future we can combine it also with some soil proximal sensing that it can help us for the detection of the appearance of this deeper loamy layer. And there's a potential of the use of the agroecosystem models to simulate within field heterogeneities and their impacts in crop response. So for the next steps, we would like to continue testing the model for the optimization of crop and field geometries for the delivery of the ecosystem services. We will also like to improve the model in terms of the quantification of biodiversity indicators. So how to try to identify options to try to quantify these effects of the diversified cropping system and new field arrangements in terms of biodiversity, and also some data assimilations in terms of that can be used as an input to the model for model improvement, and also the improvement of the spatial of the simulation of the spatial heterogeneities in the soil. So with this, I finish. So as a final outcome, we expect to gain mechanistic insights into the scale-dependent effects of crop diversification on agroecosystem performance, and what we aim also in the long term is to develop and apply agroecosystem models to study, but also to optimize these diversified cropping systems at the local and the landscape scale by considering different trade-offs in terms of not just productivity, but also the delivery of ecosystem services, biodiversity, and possibly also economics. And with this, I finish. Thank you all for your attention, and I'm open for questions.